Yes, this is the sixth part of chapter Principle of Inheritance and Variation. In the previous part 1, 2, 3, 4, we have covered about Mendel, father of genetics, role of Mendel, role of pea plant, different laws given by Mendel, and some terminologies that are used, and exceptions of law of dominance, that is codominance or incomplete dominance. So monohybrid growth also. So let's start. Now, this is the dihybrid cross. This topic will be considered in dihybrid cross, and heading of this topic is inheritance of two gene. In the previous parts, only single gene is considered for crossing. Only single gene, that is monohybrid cross, is considered. But in this slide, in this topic, we will cover with two genes at a time. So this is the uh, law of independent assortment. Law of independent assortment. What is the meaning of assortment? Assortment is arrangement. Assortment is arrangement. So let's start. Definition of law of independent assortment is factor for different pairs of contrasting character in a hybrid. Means if an individual contain two character, two contrasting character, then these two character will separate independently or distributed independently during the gamete formation. During the gamete formation. So I'm again repeating. Law of independent assortment states that when in an individual there are two contrasting characters were taken, then these characters will separate or assort independent during the gamete formation, and they independently rearranged. They independently rearranged, and this Mendel's second law was explained with the help of dihybrid cross. What is dihybrid cross? When the cross is made between two parents but having the two pairs of contrasting character, having the two pairs of contrasting characters. This is dihybrid cross. We are taking the two characters simultaneously. First is the the seed color and the seed shape. Two characters one time. So round is the shape, yellow is the color, wrinkle is the shape, and green is color, seed color. So round is given with the genotype capital R, capital R. As we know, round is dominate over wrinkle, and yellow is dominate over green. So we have to write with the capital symbol, that is capital R, capital R, capital Y, small r, small r, small y, small y. These genotype will produce gamete, that is from capital R will be taken, capital Y, only capital Y will be taken, and small r, and small y, small y. So these are the gamete produced, and this gamete will fuse to form, these gamete will fused to form F1 generation, and these are capital R, capital Y, small y. And as we know, capital alphabet will represent or dominate over small. So capital R will express as a round, and capital Y will express as a yellow. This is the first generation result. All the individual will have round yellow color, but we have to solve till second generation, and second generation is obtained in the previous slide. Also we discussed about second generation is produced by crossing first generation, that is F1 and F1. F1 and F1 is crossed to produce second generation. Yes, you can see here. This is parental generation. This is also parental second, and this parental will combine to form first generation, and first generation will produce gamete, and there are four types of gamete are produced. This is capital R, capital Y, capital R, small y, small r, small r, capital Y, small r, small y. These four gametes are from first parent, and below these are also four gametes by second parent or second after generation, and these are crossed to produce the result. These are crossed to produce result. You can see in the box. It is very easy to draw this Punnett square. The method is known as Punnett square. Uh, how we draw this? Just imagine you are in box. Just imagine you are in a box, and above this is capital R, capital Y, and sidewise capital R, capital Y. So assemble these, then this expression will be there. Then again you have to imagine and see upside, that is capital R small y and capital R capital Y from this side. So by this method you draw all the 16 combinations, and by identifying this 16 combination we obtain the ratio, that is 9 ratio 3 ratio 3 ratio 1. We have to identify this and we have to solve this. Yes, this is dihybrid test cross. What is test cross? When F1 hybrid is crossed with its recessive parent is known as test cross. So in dihybrid test cross, F1 hybrid is crossed with its recessive green wrinkled pea plant, and recessive green wrinkled is expressed with the phenotype small r small y, and the gamete will be small r small y, and this F1 hybrids are crossed to form or to have the phenotype ratio, that is 1 ratio 1 ratio 1 ratio 1, 4 times 1 ratio will be there. This is the phenotype ratio of the dihybrid test cross. Thank you. This is all about in today's lecture.